Hi everyone, my name is Morgan, and for my experiment in bio, I decided to test the effects of millipedes on soil acidity. Millipedes are important decomposers in many ecosystems due to their role in nutrient cycling and breaking down materials. They may have a profound role in directly changing the soil acidity or pH, since nutrient cycling is one of the key processes of ecosystem dynamics. Studies in the past have shown that changes in soil elements and acidity are associated with rehabilitating areas, so millipedes may play a key role in rehabilitating the soil and areas of an ecosystem. The ivory millipede was used in a microcosm approach to study the changes in soil acidity in different densities. I chose this species mostly because I already have them at home. My hypothesis was that with a higher density of millipedes, there will be more activity within the soil, and more of the soil will be synthesized by the millipedes. Consequently, my prediction was that if there is a higher density of millipedes, then the result will be an overall lower soil pH. To run this experiment, I set up 4 different plastic containers that were 13 inches by 8 inches by 5 inches. All contained organic topsoil from a local gardening store, magnolia leaves, sphagnum moss, and cork bark to allow millipedes to feed. Each container had a different density of millipedes. I decided to do 0, 2, 4, and 6 because that is how many millipedes I had available. They were all kept at room temperature, which is approximately 72 degrees Fahrenheit, and misted with dechlorinated water daily to keep the proper humidity up. Every other day, data was collected using a digital rapid test soil pH measuring device. Data was collected for a total of 3 weeks. Data was then inputted into an Excel file, and changes in soil acidity were analyzed in R. A scatter plot of the data allowed further visualization and granted further analyzation of what occurred during this study. In Figure 1 shown here, the influence of millipedes had a greater effect on the overall soil pH over time. We can see a downslope and trend line for each of the groups with millipedes. The slope is larger in groups containing more millipedes. The control, which contains 0 millipedes, can be seen with a pH of about 8.0 throughout the duration of the experiment. Based on this graph, there is a significant relationship between the day and soil pH depending on the density of millipedes. The soil acidity of containers with the larger density of millipedes was significantly lower in pH than the soil in containers with a lesser density of millipedes. I found an F statistic of 30.52 with degrees of freedom of 1 to 42 and a very small p-value. With such a large F statistic, this shows that the variation among density means is more than what you would expect to see by chance if the null were true. A small p-value also provides evidence against the null hypothesis that there is no relationship between soil pH and millipede density. This data does support my original hypothesis that a higher density of millipedes will result in a lower pH over time, due to more soil being synthesized. Similar findings to Gonzalez, Murphy, and Bellin's study, who found that the presence of millipedes caused the pH to slightly but significantly decline when they were exposed to soil in higher densities. A longer experiment in this case might have seen more dramatic results. The type of soil might also play a role as well, along with the level of nitrogen in the soil. Nitrogen is important to note because if the percentage of ammonium increases in soil, possibly due to fertilizers, the acidifying potential will increase as well, consequently lowering the pH. These findings are important regarding soil regeneration and the flow of nutrients to help plants grow. A study done by Seth and Bhatnagar using soil culture experiments found that there is a linear relationship between water holding capacity and soil pH. Most plants will thrive at a pH of about 6.5, and in soil with a larger holding capacity. Millipedes have the potential to help soil reach those levels and to help plants thrive. The introduction of millipedes in areas that require soil regeneration in order to thrive may be beneficial. As far as further research, the interactions with millipedes and other arthropods in their natural environment may affect the influence on soil pH. Millipede impact on decomposition processes and soil acidity may vary depending on the species. Since this study only used one species of millipede, it may be beneficial to experiment how other species affect the soil pH as well. Further studies on this topic may also want to hold an experiment with a longer testing time, along with using different types of soil, as they may play a vital role in affecting the soil pH. This slide shows the primary sources used in my research to help conduct my methods as well as viewing my results. Thank you so much for watching and have a great day.